Now, this is a complex table, nonetheless, and it's also not one that's necessarily widely accepted scientifically. This is my personal set of definitions about how I view biomarkers in drug development, and that includes separating pharmacogenetics and pharmacogenomics. A lot of investigators do not separate them and call them both pharmacogenomics. By pharmacogenetics, I'm referring to the germline native DNA sequence of an individual, not the disease tissue, but the healthy cell's inherited DNA sequence. The pharmacogenetics type research is often called SNPs, SNP discovery, polymorphisms, single nucleotide polymorphisms, and multiple, and genotyping. In other words, determining whether the profile sequence in the native DNA of a patient will influence the efficacy and toxicity of a pharmaceutical agent. Generally, this has had some prediction of efficacy. One of the most recent dramatic ones is the SNP polymorphism for the cytochrome P450 gene, the 2D6, associated with complete resistance to tamoxifen therapy, hormonal therapy in breast cancer, related to the fact that a small percentage of patients lack an enzyme needed to activate tamoxifen after the patient ingests the tablet. So they circulate an inactive form of tamoxifen, and the biomarker could be genotyping, but another biomarker for that would be a premenopausal woman takes the anti-estrogen and doesn't experience any menopausal symptoms. The oncologist immediately should be alerted to that, say, my goodness, this is a 2D6 polymorphism, tamoxifen's not the right drug for this patient, she can't possibly benefit from it, she'll never activate it. But we can also genotype the patient before the drug is given and predict that. Pharmacogenetics is particularly useful for the prediction of toxicity. And we see specific genotypes involving metabolism associated with all sorts of side effects from various drugs, cancer drugs in particular. Pharmacogenomics is the messenger RNA expression, the profiling of the human genome, 40,000 genes, usually of the disease tissue, not of the patient's normal germline cells, but the cancer cells, for example, the synovial cells in rheumatoid arthritis, the intestinal epithelial cells in inflammatory bowel disease, the endothelial cells in cardiovascular disease. Specific for the disease, this is most effective at deciding drug resistance or prediction of efficacy, and not particularly good for predicting toxicity. Toxicogenomics is a form of RNA expression that is designed to predict toxicity. Metabonomics is a modeling, a mathematic and statistical modeling set of data for each patient designed to predict the response to therapy based on the metabolism of the agents, and thus to get the dosing and route of administration right. And pharmacoproteomics is the latest to come on, the least well developed, where instead of the gene expression signature, we look at the protein production signature and try to predict either toxicity or efficacy using protein research rather than gene research. Regardless of what technique we use, it can occur throughout the entire life cycle of drug development. Here we see for SNPs a chart showing they can be used to identify appropriate targets, to validate the targets, to develop the lead compounds most likely to be efficacious, to evaluate the preclinical models, to check for the efficacy in the early clinical trials, and then post-market be used to select patients who are most likely to benefit or least likely to achieve toxicity of the newly formed agent. In other words, using the polymorphisms to guide which patients get the drug and which do not. And finally, in the last about minute, I'd like to talk a little bit about other forms of biomarkers, because they're not all blood tests and tissue tests. Imaging and radiology is a form of biomarker. Here's what you can see are four sleeping mice who've actually got a form of a cancer placed in their hip pad here. We call this the xenograft model. And what we've done here is construct the cancer cells to be able to give out a fluorescent signal that can be read by a special camera to predict whether or not the target of a drug is actually being hit. And without going into great detail, here's the cancer in full effect, with red being the most intense signaling. And here, after the drug is given, you see how we're able to turn off the fluorescence of the reporter system, indicating that the target of the drug has been inhibited. We can use this for dosing and for proof of principle that in vivo, just like in vitro cell lines, for example in cancer, that drugs are actually able to hit their target. Whether we can ever do this in humans remains to be seen, because we have to ingest this naturally fluorescent protein, which is currently not FDA approved for human consumption. Also, the human is a lot thicker creature, and the camera sensitivity to get the imaging through the human skin is not nearly as easy to do as it is in a very thin-skinned, white furry creature like we just saw. But we do have attempts to try to do this in human. Here's an example of a type of imaging called annexin 5, in which we are able to show, as you look at this patient with this particular cancer image here, that as we give a therapy, we can show changes in the functional imaging, predicting that the tumor in the patient is actually responding to the drug. And of course, PET scanning is widely used now to predict response to therapy, and one would predict that imaging modalities will continue to be part of biomarker-based drug discovery, but these would be what we call imaging biomarkers. So this is the end of my introductory presentation. For those of you who select the scientific breakout session, I'm going to talk about a series of biomarker-developed drugs, specifically in the cancer area, and those will include the HER2 trastuzumab or Herceptin story, the BCR-ABL and imatinib or Gleevec story, and a couple of others like that. And I look forward to joining those who come to that in a little while. Thanks very much.
Biomarkers have the ability to help you improve cost efficiency, predict and minimize risk, avoid late-stage attrition, and make better, more informed decisions. BIOMARKERcenter from Thomson Reuters, the world authority on the pharmaceutical industry, is a single, comprehensive resource, providing you with the information you need on biomarkers at all stages of development.